Hey there, this is your fourth of 5 fully vodcasted lectures, and if you're joining me from the classroom today, then it is Wednesday, and we're looking towards the week of spring break coming next week, so I bet you guys are jazzed about that, and that also means that I don't have a lot of really poignant announcements to make today. That is to say that we'll be looking ahead towards homework 7, which will be due on the week after spring break, and that'll be Tuesday, March the 20th. However, looking even further ahead than that, the week after spring break is going to be your third exam, and that's the exam for which you're going to want to have your stunning metabolism poster all done and ready to go, and hopefully ready to be an amazing resource for you. Remember that you can also include on that poster all of this information about gene expression. So if you have a creative way of picturing that, the rules on the poster, of course, are that there can't be any blocks of text. It must be of your own making, and it needs to be on a single sheet. So however you want to do that, whether it is literally a sheet or whether it's a large poster board or something like that, it is a single sheet that I'd like for you to have that poster. I don't have much in the way of announcements for lab tomorrow, only that you can be looking forward to more biochemical tests, and please be good to your TAs and to Cassie tomorrow again, as I'm sure you were last time. We're going to go ahead and jump right back into where we were talking about the elongation phase of translation. And when we had left off, we had just seen how initiation takes place, where all of the complexes can come together at the initiation site, the 30S ribosomal subunit binding to the Shine-Delgarno sequence, and then the initiation factors helping to allow, initiation factor 2 helping to allow the binding of the N-formal methionine carrying tRNA. And eventually, once everything was together at that initiation site, the 50S ribosomal subunit came in, we saw hydrolysis of GTP, and finally we saw the whole ribosome put together and ready to head off in elongation. So that's where we're headed today, is to look at elongation, which really can be split down into 3 parts as well. Essentially, the first part of that process is the binding of the tRNA to an open site. The second part of that process is going to be the synthesis of the peptide bond. And then finally, the third part of the process is going to be when the ribosome moves along. We call that translocation, that it kind of heads on down to the next codon on the mRNA. So before we jump into the actual elongation process, I want to point out a few sites on the ribosome. These sites are pretty easy to remember as a whole. One of them you already know, that is the mRNA binding site. Remember that within that 16S ribosomal subunit, there is a pyrimidine-rich region that recognizes that very purine-rich Shine-Delgarno sequence and allows that 30S subunit to really align with the right spot on the mRNA. And of course that alignment situates it so that the start codon is in the right spot to bind to the initiator tRNA with its F-met. So we'll recognize a couple of other sites, one of which the tRNA with the F-met will be bound at the very beginning. That is the P or peptidyl site. That's a mouthful. And that P site is going to be the site that the tRNA carrying the F-met will be right after initiation. But there's also a second site to the right of the peptidyl site, and that's the aminoacyl site. The A site is the open site where the next tRNA carrying the second amino acid in the chain will be able to bind. Now to the left of the P site is the E site, and of course that stands for exit. So that means that as the ribosome moves downwards, down the mRNA, it's going to pop the tRNA that was in the P site into the E site, and then that tRNA will leaving behind, of course, the amino acid that it was carrying. So that'll keep happening again and again. Okay, so the EP and A sites. Now, some of you may have been looking at me and thinking, wow, is Rachel wearing a ribosome today? If that's what you were thinking, then you are spot on. So I'm going to see if I can get up and show you my ribosome t-shirt. Now, I have been yearning for this ribosome t-shirt for a long time, and finally last semester one of my students made it. So you can see that I have my E, P, and A sites, and notice that the tRNA that is bound to the amino acid, the current amino acid, is in the P site. It's recognizing on the mRNA, look at my mRNA, pretty sweet, Okay, on the mRNA being read, here's my 5' end of my mRNA, and here's my 3' end. So the ribosome is reading in the 5' to 3' direction, and as soon as the this tRNA is bound in the P site, and as soon as this A site is empty, a new tRNA will bind into that A site. And then as the whole ribosome moves along, then what will happen is that this tRNA will pop into the E site and leave, and it'll of course leave behind the polypeptide chain. And you can see my ever elongating polypeptide chain there. One of my students named Kendra is the one who made this awesome shirt for me. So I hope it helps you visualize as we go into the next step, in which we see sort of a similar picture where you can see that the P site is currently occupied by the N-formal methionine carrying tRNA, and the A site is open. So that would mean in the A site, this, what I'm showing as purple right there, is actually a codon. That codon, you know, it could be anything. It could be AGU, for example, meaning that the next tRNA, if that was an AGU codon, the next tRNA would carry serine on it. So that serine carrying tRNA would pop into the A site. And we know that the next step would have to be the formation of a peptide bond between the F-met and the serine on that second tRNA. But we'll get there. Let's write that down before we get way too excited and run along. So we recognize that the F-met is bound at the P site, leaving an open A site, and the second tRNA carrying its amino acid can bind at the A site. Now, at this point, the second amino acid we had maybe made it up to be serine, that would bond via peptide linkage to the F-met. Remember that the 23S rRNA is actually what catalyzes the formation of that peptide bond, right? Wicked cool enzymatic activity coming from an RNA there. So let's see that happen. We sometimes call that step transpeptidation. So if you're in for the sexy-minded term, that's what we're looking for there, is the formation of that covalent bond. This is called transpeptidation because it's the formation of a peptide bond linking the two together. That means that the ribosome is literally ready to move along. So it's going to move down in the 5' to 3' direction this way, and in doing that, it's going to cause the tRNA that's currently in the P site to go into the E site. And once that tRNA goes into the E site, it's going to make its exit. So that'll be what happens in the next step, and this is called translocation. Now, it's worth noting that this, again, for a second time, this is very energy intensive, right? There's going to be a lot of hydrolysis of high energy bonds to power translation. And certainly every time a new tRNA binds in the A site, there's going to be an elongation factor that's going to help to guide that and to lead it into there. So there's a lot of things involved in this that we're not showing, but we're getting the rough idea of what's happening as the ribosome heads down and reads the mRNA. So clearly translocation, after that happens, it frees up the A site again. So that means that the third amino acid carried by its tRNA can come into that A site then at this point. So after translocation, we're back again to the beginning, binding of a new tRNA, transpeptidation, and then translocation once again. So here we see momentarily vacant A site because that second tRNA will occupy the P site, but in very short order, that third tRNA will come in and bind to the A site. So very quickly, that A site will become occupied by the third amino acid. Now, that could be that what's shown in yellow here is another codon. Maybe it's CGU. You guys can look up and see what maybe that would encode there. So this is the repetitive process, right, that happens as the ribosome moves down the mRNA and continually adds a new amino acid to the constantly elongating chain. And we, of course, remember that the first terminus to be exposed here, once it does pop out, will be the amino terminus. So the weird thing about this is that once the whole translation machinery has left the Shine-Dalgarno sequence and has kind of cleared out and headed down the mRNA, that means that it opens up the Shine-Dalgarno again to bind to another ribosome and for the initiation complex to all form again and the whole jazz to take over again. So this can happen simultaneously, where you can get more than one ribosome bound to a single mRNA at any given time. And we actually have a name for that. It's called the polyribosome, which makes sense, or for short, little contraction, the polysome. So the polyribosome or the polysome, more than one ribosome reading the same mRNA at the same time, right? That's intense. So now we recognize, after many steps of heading on down that mRNA transcript, eventually what pops into the A site is what we call a nonsense codon or a stop codon. So that stop codon doesn't encode for any amino acid. No tRNA to swoosh into the A site, and in fact what will happen at this point is release factors will play a role in releasing the polypeptide chain and now allowing it to fold into its full three-dimensional structure as a protein. So proteins called release factors play a role in that. And of course, just for good measure, make sure to label your amino terminus, right? It would be the same on my t-shirt, and I can't remember if Kendra did this or not. I think she just put an F-met up here, but that would be the amino terminus that would be up on my upper right shoulder. So we recognize now, once the ribosome has hit that stop codon, the polypeptide chain has been freed and it's folding, that now the ribosome falls off of the mRNA and it dissociates into its components. It dissociates again into its 30S and 50S subunits. Again, those initiation factors might associate as it starts another round of translation somewhere else, or on a similar mRNA, or on a different mRNA. So those will dissociate at this point. Okay, so that's termination of the process, and I think we got through all of the 3 steps and are feeling hopefully pretty good about that. However, one quick note is that sometimes after a polypeptide chain has been released, there are sometimes processes that still occur, that is post-translational processes, right? So many proteins are processed post-translationally. And one of the things that generally tends to happen there is that the first few, maybe 3 or so of the first amino acids in the chain are actually clipped off. So if you've ever seen a protein sequence for a bacterial protein and it didn't start with F-met and you were like, whoa, what's going on? How come there's no F-met at the beginning there? The reason for that is because of post-translational cleaving of a few of the amino acids off of that chain. There are other post-translational processes that can also occur. For example, a signal sequence might be added. Remember that that would be important for those proteins that would be exported via the sec machinery. Or we might see that a chaperone may come and help a protein fold. That's another protein that provides a safe kiva for folding of the protein. And finally, of course, we see that cleavage of 1 to 3 amino acids at the amino terminus being removed, and always including the N-formal methionine. So most protein sequences that will read will not actually read beginning with the N-formal methionine. Awesome. I guess if we were in the lecture hall right now, I would ask for questions. But I guess what I'll ask you for is to please email me if you have any questions at all about initiation, elongation, termination of the translational process.